Hello everyone, my name is Vivian Zhang. I'm the product manager of cell biology at GenScript. Today I would like to talk to you about the stable cell line catalog products for immune checkpoint drug discoveries. So here is a list of the contents I would like to go through with you today, including a brief introduction of the history, the drug discovery cell line generation, our portfolio, highlights, summary, and some technical supports. So here is a very brief introduction of the histories. As you may know, GenScript was established in 2002 in New Jersey, USA. We're now a leader in the biotech. In 2009, we started to provide GPCR stable cell lines for drug discoveries. 7 years later, in 2016, we expanded our portfolio to provide also immune checkpoint stable cell lines. In 2017, we further expanded our portfolio to include some Fc receptor stable cell lines. So far, we have successfully developed more than 50 stable cell lines for immune checkpoint drug discoveries. So here is a brief introduction of the workflow of how the cell lines are generated. We started with the plasmid construction, which include the target gene as well as a selective gene. And then we performed the transfection, followed by antibiotic selection. From here, we got the polyclonal stable cell pools. And then we performed the dilution cloning to get a single clone stable cell line. And then each of the clone is further expanded individually to select for the best one to be our product clone. The product clone was then banked in our inventory, further expanded for harvesting, and a portion of the cells were used for QA before the cells become deliverables. So the first topics in the cell line generation was host cell lines are chosen. All of our host cell lines are chosen for the best performance of the cell lines for drug discovery performance. So far, we have 4 different options. CHO KI cell lines are one of the most popular used in biotech. These are the epithelial cell lines derived from the ovary of Chinese hamster. The strain we used here is a DHFR sufficient strain. These cells are easy to culture, they're tightly adherent, low cost to grow with simple medium. We also use some HEK293 cell lines, which are derived from the human embryonic kidney cells. These cells are widely used due to their reliable growth as well as the propensity for transfection. Different from the CHO KI, the HEK293 cells are compatible for different platforms, especially for human disease targets. However, they're easy to detach, therefore, they mostly grow out in suspension. HT1080 is another cell line that we use for the reporter cell generation. They are derived from the human fibrosarcoma biopsy. We found it as a suitable model for NF-kappa B signaling pathway, which is necessary for immune responses. Jerkit is another cell line that we use for generating the reporter cell line for immune checkpoints. They are a human T-cell cell line, therefore perfect model for in vitro assays to mimic the real immune responses. Talking about target genes, there are a couple efforts that we have done in our cell lines to ensure the best results. It has been found in recent research that the using of labels may cause artifacts in the drug discovery results because the label may interfere with either the compounds or the drug targets. Therefore, non-tags are used in our cell line generation. The non-tag technology allows for the label-free investigation, therefore guarantee higher accuracy in the drug discovery. Another condition that is frequently faced with is the gene expression levels are sometimes limited due to the functionality of the gene or the host cells. In such conditions, we apply codon optimization technology according to different host cell lines in order to increase the gene expression level when this is necessary. In order to select the clones with the gene of target, we use the antibiotic selection systems. This including multiple different genes that are resistant to antibiotics, for example, the hygromycin B, or pyrimycin, or zeaxanthin, or geneticin. As you can see in the diagram down here, the antibiotic resist genes is inserted in the plasmid, same plasmid as the target gene is, and also enter the same promoter to drive the target gene expression. Therefore, only the cells that is inserted with the target gene will become antibiotic resistant, therefore can be selected out from the cells that do not have the target gene. Two different types of clonal screenings are performed in our cell generation. The first one is to screen for the single clone. The cell clones that we got after the antibiotic resistant selection is actually highly heterogeneous. There are cell pool instead of a single clone. This is because of the random integration of the foreign genes into the chromosomes. Therefore, we perform dilution cloning to select for the single clones, where we dilute the polyclonal cell pools and implant them into the 96 well plate to ensure that no more than one cells per well. And then after a couple days of expansion, the wells with a cell will become expanded into maybe hundreds or thousands of the cells. And here you get a clone from one single cell. All the single clones are then further proceed for the screening to select for the product clones based on the best performance. The highly producing clones are actually rare because the active region supporting the high gene expression in the chromosome is rare. Also, some immune checkpoint proteins can actually induce apoptosis. Therefore, we use FACS assays to screen for the clones for binding assay cell lines, and we use functional assays to screen for the clones for functional assay cell lines. Next question, you may wonder, where can the cell lines be used in the procedure of drug discovery? Here I'm showing you the procedure of the whole drug discovery. As you can see, it involves many different steps. Actually, it can be sectioned into three major steps. From the target discovery all the way to the lead optimization in the first row, this is the drug discovery. After that, starting from the development and then including the IND, this is your drug development section. And with a good result from the clinical trials, you'll be able to apply for a new drug application and bring the drug into the market. This is your drug commercialization stage. Within the drug discovery stage, there are two steps. After you identify the drug, after you identify the target and validate the target, and now you may have thousands of compounds where you need to screen for the hits, which are the compounds that can bind or show some affinity to the drug target. This is where the overexpressing cell lines for binding assays will come into place. And with the hits found, next step you will need to find the leads, which are the compounds that not only show binding, but actually show some functionality to the target. For example, either increase or decrease the expression or to inhibit the drug target. So this is where the reporter cell lines for functional assays will take into place. In our portfolio for the immunocheckpoint stable cell lines, we have both cell line types for the two different stages of the drug screening. We have 50 overexpressing cell lines for the binding assays, including species for human, mouse, and dogs, as well as 7 reporter cell lines for functional assays. All the species are humans here. With such extensive portfolio, we are able to cover almost all the popular drug targets for immune checkpoints, as you can see in the picture in the right panel. We're also continuously developing new products to facilitate the research on the newly discovered immune checkpoints. Our products are highlighted with the 3 features listed here. They're stable in passages, they present high SB ratio, and also they're ready to use. Firstly, I would like to talk about the stability. As you know, the reliability and data reproducibility is very important in drug discoveries. Therefore, all of our cell lines for drug discoveries are tested for stability for at least 15 passages. Down here, I'm showing you example data of the stability test in the HT1080 OX40 reporter cell lines. The cells are tested for stability after passages for 1 time, 5 times, 10 times, 15 times, or 20 times. As you can see, the expression of the L8, which is the assay readout, is quite consistent in between cells after different passage numbers, showing very high stability. The second feature that is very important is a high signal background ratio. FACS assays are performed for the signal background ratio in the cell lines for binding assays. In the left panel, I'm showing you the data of the CHO KI, PD-1 stable cell lines. As you can see, the expression is detected by the FACS assay in the CHO KI whole cells in red and the CHO KI PD-1 stable cell lines in the blue. As you can see, the 2 peaks are very distinct from each other, indicating very high SB ratio. Compared to this, on the right side, I'm showing you the FACS assay of the competitor's FD1 stable cell line. As you can see, the signal for the negative control and the signal for the stable cell lines are very close to each other and actually overlapping, indicating a poor SB ratio, and this could be an issue in the drug discovery experiments. Another example is the SB ratio test for the cell lines for functional assays. Here I'm showing you the cell line of HT1080 OX40 stable reporter cell lines. With OX40 ligand added into the cell culture, the L8 expression levels are detected. As you can see, the signal of the L8 expression from the OX40 cell lines showing in blue is significantly different from the signal from the parent cells, which is the whole cells that is shown in red, indicating a very high SB ratio. So the last feature I would like to talk about that is also very important is the ready to use. As you can see, the whole procedure of the cell line production takes about 20 weeks. Therefore, the cell line generation is so time-consuming, but the drug discovery is very competitive time-wise. Therefore, you want to speed up your drug discovery, and this can be done by taking advantage of the ready-to-use cell line product. So far, I have introduced to you our products of cell line tools for the drug discovery. We have 11 years of experience in generating stable cell lines. We have successfully developed more than 50 immune checkpoint drug discovery cell lines. They are single clone validated for high expression or functionality, stable in passages, high SB ratio, and ready to use. Therefore, they're great tools to speed up your projects and enable highly reproducible data. If you have any other question, please go into our website and you can find more information under the catalog products. And from the tab of the document, you will be able to find the FAQ information. With that, thank you for your time, and please contact us if you have any other question. Thank you.